Let us discuss fructose metabolism and disorders. First of all, what is fructose? It is a monosaccharide. It has keto group and 6 carbon. That is why it is ketohexose. It occurs naturally in abundance in all the fruits. Sucrose is a disaccharide which is made up of fructose and glucose. Sucrose is a non-reducing sugar. You can watch this video to learn why sucrose is a non-reducing sugar. Inulin is a polysaccharide which is made up of not glucose, made up of fructose, right? Fructose is not essential in diet. That means there is no need to take fructose every day. That means fructose can be synthesized by human body. First of all, why fructose is important? It is the main source of energy for sperm. Glucose cannot be directly isomerized to fructose in human body by any enzyme. There is no enzyme that will convert in one step glucose to fructose. So we need a pathway that will convert glucose to fructose. That pathway is called sorbitol pathway. Okay. Before studying sorbitol pathway, let's compare the structures of glucose and fructose. Both are isomers. They are functional isomers. Here the functional group is aldehyde group. Here the functional group is ketone group. Right? So this is C1, this is C2. So glucose and fructose, if you see, they are different in both C1 and C2. That is why you cannot convert glucose to fructose in single step. So the polyol pathway is there, in which what happens, there is an enzyme called aldose reductase, which is going to reduce C1, right? So the first carbon is reduced to alcohol. So the resultant product is known as s orbital or a polyol. Why it is known as polyol? OL means alcohol group. See this? There are 6 OH group in sorbitol. Sorbitol is not a carbohydrate. What is the definition of carbohydrate? Polyhydroxyaldehyde or polyhydroxy ketone are compounds which produce them on hydrolysis. Sorbitol neither has a aldehyde group nor a keto group. So it is not a carbohydrate. It is a polyalcohol. Right. So now in the next step, what are we going to do? We are going to convert this alcohol group into ketone group by the enzyme sorbitol dehydrogenase. So we get fructose. Clear? Very simple, right? So polyol pathway converts glucose to fructose. The enzyme involved are aldose reductase, sorbitol dehydrogenase. Why is the name aldose? Because glucose is an aldose. It has aldehyde group, right? So that is why aldose reductase. I have discussed the role of sorbitol in diabetic cataract in this video. You can watch the video later. So having discussed the polyol pathway and all, let us now go and discuss fructose metabolism. Then we will discuss the disorders. Fructose is catabolized by a pathway called fructolysis. So similar to glycolysis, this is called fructolysis. It takes place mainly in the liver. There is an enzyme called fructokinase and there is an enzyme called aldolase B. Let me make things simpler. Look at this. The first enzyme is fructokinase, which converts fructose to fructose 1-phosphate, not 6-phosphate. Okay. So this mainly takes place in the liver. Next, there is an enzyme called aldolase B. This is not aldolase A, which is involved in glycolysis. This is aldolase B. This is also taking place in the liver. It splits the fructose 1-phosphate to dihydroxyacetone phosphate and just glyceraldehyde, not glyceraldehyde 3 phosphate, because there is only one phosphate here in fructose 1 phosphate, right? What happens next? You remember we have seen DHAP in glycolysis, right? So this DHAP can go to glycolysis. Glyceraldehyde is converted into glyceraldehyde 3 phosphate, then it enters glycolysis. So ultimately, fructose enters the glycolysis. So initially, there is a pre-processing called fructolysis. Finally, fructose enters the glycolysis. The problem is that fructose enters the glycolysis bypassing the rate limiting step, that is PFK1. Okay, remember this. This is very, very important point. So it enters glycolysis bypassing the rate limiting step. So the fructose metabolism is not regulated. All right. So that is why diet high in fructose causes hypertriglyceridemia. So there will be unnecessary glycolysis happening. There will be pyruvate, there will be acetyl-CoA, etc., etc. Okay. In addition to that, fructose also activates certain genes which are involved in lipogenesis. So that is also the reason for hypertriglyceridemia. So there will be hyperuricemia also due to excess ATP breakdown. So there will be uric acid production. All right. Now we will discuss the disorders associated with fructose metabolism. So what we have studied? We have studied two enzymes, fructokinase and aldolase B, right? So defect in fructokinase leads to essential fructosuria. Look at this. Fructokinase is absent. So fructose is not phosphorylated to fructose 1 phosphate. So it is simply excreted in the urine, right? So essential fructosuria is a benign condition. Hereditary fructose intolerance, which is due to defect in aldolase B enzyme, is a serious condition. Why? See this, in hereditary fructose intolerance, this is happening, right? Fructose 1 phosphate is happening. So there is no problem. But the next step is not happening, right? So excess of fructose is converted to fructose 1 phosphate. So there is accumulation of fructose 1 phosphate. So what happens? ATP is converted to ADP. So there is depletion of ATP and accumulation of a charged compound inside the hepatic cells. Okay. So remember this. Next, what is going to happen? This fructose 1 phosphate inhibits glycogenolysis. So it inhibits the glycogen breakdown. So if glycogen is not broken down when it is needed, what will happen? There is chance of hypoglycemia, right? Look at this. There is ATP depletion, I told you, right? Because of this, there will be low ATP and there will be high ADP. Because of low ATP, gluconeogenesis will be inhibited. Conversion of two molecules of pyruvate into one molecule of glucose requires 6 ATP equivalents. So because of low ATP and inhibition of glycogen breakdown in hereditary fructose intolerance, there will be hypoglycemia. Remember this, very, very important point. So why there is hypoglycemia? There are two reasons. All right, now focus on this high ADP. So this high ADP will go to this reaction. ADP reacts with another ADP to produce one ATP and one AMP, right? So this is your substrate level phosphorylation. The name of the enzyme is myokinase or adenylate kinase. So here there is a lot of AMP production. So this AMP will be finally degraded to uric acid. So in hereditary fructose intolerance, there will be hyperuricemia. hyperuricemia. Okay. So there is hypoglycemia, hyperuricemia. In essential fructosuria, only fructose is accumulating. It is a benign condition. Whereas hereditary fructose intolerance, it is a serious condition. So there is liver damage because of accumulation of fructose 1-phosphate inside the hepatic cells. So it causes toxicity. There will be hypoglycemia, hyperuricemia, right? So these are the major clinical features of hereditary fructose intolerance. This liver damage ultimately will lead to cirrhosis. Okay. So the children with hereditary fructose intolerance, they will learn to avoid food items that contain fructose. So these children, they won't eat fruits at all. Right? Final question. Can diabetics use fructose as a sweetener? Think. In diabetic people, blood glucose level is high. What if they think that, okay, my blood glucose is high. Instead of glucose, let me take fructose. What will happen? It will lead to hypertriglyceridemia and hyperuricemia. ICMR guidelines for management of type 2 diabetes says like this. Nutritive sweeteners. These include fructose, honey, corn syrup, molasses, fruit juice, or fruit juice concentrates. So all these are best avoided. Okay. I hope you have learned fructose metabolism and disorders. If you have any doubts, feel free to ask me in the comment section. Thank you.